Good afternoon everyone, welcome to my channel. I'm Sharmin. Today I'm going to teach you biology, so let's get started. Action potential, this is the topics I'm going to talk about. So yeah, with a small addition to the apparatus shown in here, so you can see here the diagram, and it's possible to stimulate the axon with a very brief and small electric current. So if the axon is stimulated in this way, the steady trace on the computer screen suddenly changes, right? And the potential difference across the cell surface membrane and of the axon suddenly switches from minus 70 millivolt to plus 30 millivolt. Okay, so it swiftly returns to normal after briefly overshot. Okay, so the whole process takes about 3 milliseconds, and this rapid fleeting change in potential difference across the membrane is the action potential. It's caused by changes in the permeability of the cell surface membrane to sodium ions and potassium ions. Okay, so you can see here the diagram, then you understand easily. As well as the channels that are open all the time, there are other channels in the cell surface membrane that allow sodium ions and potassium ions to pass through. They open and close depending on the electric potential voltage that is across the membrane, and are therefore said to be voltage. Voltage-gated channels. When the membrane is at its resting potential, these channels are closed, and first the electric current used to stimulate the axon causes the opening of the voltage-gated channel in the cell surface membrane, and which allows sodium ions to pass through. Okay, so as there is much greater concentration of sodium ion outside the axon than inside, and sodium ion enter through the open channels, and to begin with, only a few channels open. So these channels the potential difference across the membrane, which becomes less negative on the inside. So this depolarization triggers some more channels to open so that more sodium enters. So you can see here the diagram. So there is more depolarization. If the potential difference reaches about minus 50 millivolt, then many more channels open and the inside channels are potential. Okay, plus 30 millivolt compared with the outside. So this is an example of positive feedback, because a small depolarization leads to greater and greater depolarization. Okay, so yeah, and action potentials are only generated if the potential difference reaches a value between minus 60 millivolt and minus 50 millivolt, and this value is the threshold potential. If it's less than this, then an action potential doesn't occur. Okay, after about 1 millisecond, all the sodium voltage gated channels close, so sodium ion stop diffusing into the axon. So at the same time, the potassium ion channels open, and potassium ion therefore diffuse outside of the axon, so down their concentration gradient, and the outward movement of potassium removes positive channel from inside. the action to the outside, and this returning the potential difference to normal, and this is called repolarization. Okay, so in fact the potential difference across the membrane briefly becomes even more negative than the normal resting potential. The potassium ion channels then close and the sodium ion channels become raspier. So the potassium ion channels then close, the sodium ion channels become responsive to depolarization again. The sodium and potassium ion pump continue to pump sodium and potassium in, and this helps to maintain the distribution of sodium ions and potassium ions and across the membrane, so that many more action potentials can occur. So if you understand my topics, then give a thumbs up and share with your family and friends. If you need more class, go to my website www.educatorbiology.simplesite.com. That's all.